Hey students, we're going to finish up chapter 8 today by looking at transport in and out of the cell. One important thing here is that this is a lot of information in this particular PowerPoint, and a lot of what we cover are going to be important processes that are examples of different types of mechanisms for which things get in and out of the cell. So let's go ahead and get started. There's essentially two major types of transport. One is called passive transport, and that's a process where a molecule goes down its concentration gradient, meaning that it flows from high concentration to low concentration. This does not require energy. So that is the important hallmark of it is it doesn't require energy because the molecule's flowing from high concentration to low. This does not mean that it doesn't require a special type of protein to help. So there are types of passive transport where it's just simple diffusion. An ion or molecule moves through an opening. So this would be like our CO2 and O2 in a cell. But then you can have facilitated diffusion, which means that you need a carrier or channel protein, but it doesn't require energy still. Okay. Active transport is when you're trying to move a substance against its gradient. We want to go from low to high concentration. And so this one is going to require a source of energy. And most of the time, in the examples we'll see, is it's going to be linked to the hydrolysis of ATP, which we're going to talk about in a few minutes. Okay, there are two different types of active transport. There's primary, which is the transport is linked to the hydrolysis of ATP, or there can be secondary active transport, which is driven by an ion gradient. Okay, so for simple diffusion, this is passive diffusion of a species that's uncharged, so O2 and CO2 is a good example. And what will happen is that the molecules will flow from the side that's high concentration to low concentration. Sort of a good analogy would be water flowing down a waterfall. It's going to flow down, right? We don't see water just magically float upwards like that. So in this case, the concentration gradient drives it, and that's an energy consideration there. Facilitated transport is also moving with its gradient, but whatever it is that's moving with its gradient has to have help in the case of a protein pore, a carrier molecule, or certain proteins called permeases. So facilitated will mean still it's passively crossing with its gradient, but it needs help in the form of a protein of some sort. So, okay, now let me warn you on the next parts of the slides. These are all examples so that it's not so general. So permeases will help in the transport of saccharides, so like lactose and xylulose. Okay, and those have permeases that are proteins that will help with the transport of these sugars. There's another important point, and we're going to see it in some of the examples coming up, is that you can have cotransport, which is more than one species is somehow interacting to help each other get through the membrane. So one example will be glucose and sodium ions. And that process is called Symport because they're moving the same direction across the membrane. So this is showing the flow of glucose from the cytoplasm to the extracellular matrix. And both of the ions are flowing in the same direction. Later, we're going to study the sodium-potassium pump, and that one's Antiport because the sodium is flowing in a different direction than the potassium ion. Okay, so one of the key ideas is to ask the question, what kind of species will have trouble getting through the membrane? And the answer to that will be charged or polar molecules. So an ionophore is a type of a carrier that helps ions cross the cell membrane. So in this case, this is an example. Valinomycin is an antibiotic that acts as an ion carrier. Do you need to know the specifics on this? No, so you don't need to know this. You don't need to memorize that. But it is a great example of what are the structures of these. So, for example, the outside of the carrier is hydrophobic because that's the part that has to be in contact with the lipid bilayer. And then if it's carrying an ion, like potassium ion, it has to be charged within that pocket. So those would be negatively charged, right? Amino acids that would help stabilize the potassium ion. So this is going to be one of the themes as we look at some of these, is to understand how the structure of the carrier protein is going to be affecting its function. So in the case of a carrier molecule for an ion, the outside of it that touches the lipid bilayer has to be nonpolar residues, but the part in contact with the positive ion would have to be negatively charged. Aquaporins. These are going to be channels that allow for the flow of water. And what we'll see here is an aquaporin will span the lipid bilayer, so it's an integral protein. And one of the important parts, again, is that water needs help because it's polar, right? Very polar. And so it can't get past the nonpolar part without help, nonpolar tails, the hydrophobic tails of those lipids. So the water has to go through a protein channel, and it's going to go through it, right? Interestingly enough, then, the amino acids that will touch the lipid bilayer would have to be our nonpolar residues. And then the amino acids that will be in contact with the water, if you think about it, will have to complement water's polarity. So here with that, see here, this has the oxygen that's partially negative. It would come in contact with amino acid residues that are positively charged, perhaps something like lysine with its positively charged R group. So those are positively charged amino acids. Okay. Okay, another really important molecule that we're going to come back to this a little bit later as well is glucose, because we're going to do a whole section on how we utilize glucose for energy. That's in our next section with metabolism. But glucose is polar, so it's also going to need help crossing the cell membrane. And I think this is a good time to mention something that tends to come up when we're in person. And that is just a really important thing from now on, is to understand that we have different organs with different kinds of cells. So you have brain with brain cells, you have red blood cells, liver cells, muscle cells, adipose cells, and they all have the same common machinery because they are cells, the same ribosomes, the same nuclei and all that. But they are specialized to do different functions. So in biochemistry, when we start talking about processes, we usually have to say, hey, we're talking about this one particular type of cell. And they're not all equal in terms of our discussions in biochemistry. In biochemistry, we tend to focus on brain cells, muscle cells, liver cells, and fat cells. So for brain, that's going to be neuronal cells or neurons, right? For muscle, it's going to be myocytes, liver, hepatocytes, and adipose would be our adipocytes, those fats. So I just want to lay this out for you right now. Just I've been teaching this class a lot. I always get questions of, well, is this true everywhere in the body? Well, you're going to start to notice that different cells, since they do different things, will have sort of their own little microchemistry, biochemistry going on. Okay, so with this in mind, glucose is critical for our cells as an energy source and is super important in biochemical reactions. And how will it get into cells so that we can utilize it for energy? And so the answer to that is it depends on the cell type. So in the intestine and kidneys, there are sodium glucose link transporters, and those are called SGLTs, sodium glucose link transporters. And I just want to throw that out there as a type. We won't, I won't necessarily test you on that. We saw it on a previous slide regarding the flow of sodium ion with glucose. And you can go back and take a look at that. It was shown going the same direction, so that's why they are symporters. But each of what I'm talking about here, we could spend an enormous amount of time just on these transporters. So these are just examples for us at this point. Okay. The other one are facilitated diffusion glucose transporters, which are referenced as GLUT transporters. And those of you prepping for MCAT or any of the exams for graduate school, usually there's emphasis placed on this. And there's a paper that I have on Canvas, and you can look it up, and underneath a handout. And it's a really great paper describing all the different kinds of glucose transporters. So if you need more depth, there's a reference there for you. Essentially, what I want you to think about is glucose needs these transporters because it's polar, it can't get through the lipid bilayer. And different tissues have different types of glucose transporters. In erythrocytes, which are red blood cells, and the brain, those have mostly GLUT1 transporters. And I want also you to get excited about one thing, which is that if you notice here, let's look at this one little sentence that is really exciting. And that is that GLUT1, those are mostly in red blood cells in the brain. Those have a high affinity for glucose. And look at the next part there, low Km. So Km, I'm excited to say, helps us to understand these processes. So remember from our Michaelis-Menten discussion, the lower the Km, the higher the affinity of an enzyme, for its substrate. Now these are not enzymes, they're transporters, but the same idea will apply. The lower the Km, the higher the affinity of the transporter for glucose. So when they have such a low Km, what that means is that the brain's a great example that the brain really needs glucose to fire neurons and keep our brain signaling going. So it's going to have transporters that are very, very effective at bringing glucose into the cell. Okay. Other glucose transporters, GLUT2, that's in the liver and pancreas, it has a lower affinity for glucose, meaning that it's sort of what will happen there is that to get the liver to respond to bring in glucose, because it has a higher Km, that's going to happen when we have a higher blood sugar level, so more after we've eaten. And that's going to come back into play. We're going to see this again a little bit later in the class. So next one, GLUT3 in the brain, high affinity for glucose because of a low Km. And then in the muscle, adipose and heart, medium affinity, so medium Km. The other thing too that you should note is that the uptake of glucose is dependent on insulin, which is going to be a hormone, and we're going to come back to this. Okay. All right, so active transport, I mentioned to you, what is needed is energy. And what we're going to be talking about is adenosine triphosphate, which is ATP, perhaps, I think, probably one of the most famous biochemical molecules, aside from perhaps glucose. It has a nitrogenous base here, adenine. It has, so that's a nitrogenous base, nitrogenous meaning, if you notice who's there, nitrogen. It has a sugar, which is ribose, it's the sugar. And then it has phosphate groups. And the phosphates there, if you notice, the molecule's highly charged. And what we're going to see is that this phosphate here on the end is going to be transferred to other molecules. And when that bonding event occurs, let's say the molecule's an enzyme, that phosphorylation event, the fact that it's going to bond there is going to release some energy. Then in the meantime, what's going to happen is the fact that this is such a highly charged molecule where there's a lot of repulsions, is that's going to also help this to transfer. Okay, so, and it's going to be in that transfer event that we're going to see is going to have a large delta G that's negative. And what did we say about free energy being released? When free energy is released, it can do work. Okay, so let's take a look. This is actually, this little section here is from section 7.3. It's a very short section in your book. You're more than welcome to read it. You can probably get what you need from just these slides. It's just going into a little more depth into what phosphorylation means. I have to say that as far as processes go, phosphorylation is probably one of our most important in biochemistry. It is one of the most common ways that we control or regulate enzymes. Phosphorylation events when that phosphate's transferred onto an enzyme. That's going to cause a conformational change. And the other thing, too, that's really interesting is that phosphorylation events on an enzyme, in some cases, will activate the protein, and others it deactivates. So sometimes it makes the enzyme more effective and other times less. And we're going to see the examples of both. You know that I really like chemistry. So the next slide is going to show us which amino acid residues would actually be able to grab that phosphate. And the three major ones are going to be serine, threonine, and tyrosine. So here's a serine residue. It is going to be able to grab a phosphate from ATP, and that's a phosphorylation event. So you see how here's serine, and then the ATP was shown in the previous slide. So one of the phosphates was transferred on there. And then what's left is adenosine diphosphate. So ATP goes to ADP, and then the phosphate's transferred. When this type of transfer happens, the creation of this bond releases a lot of energy, and then you relieve the strain on ATP. That's because it was so highly charged. So you have a negative free energy being given for that process. Threonine, same thing, has the OH, so it can accept phosphate from ATP. And tyrosine has the OH, which can accept phosphate from ATP. So they all have in common that the amino acids with that OH group can be phosphorylated. Okay, so let's use this as an example of a phosphorylation event. Here, this will be an example of primary active transport. And essentially what this is the sodium potassium ATPase. Now, those of you that have taken a lot of biology, this won't be the first time perhaps that you've studied this. And also, let me just say one comment is that we are just looking at this as an example of phosphorylation events and how they affect an enzyme. This whole thing is part of the physiology of the brain. And so if you were taking a physiology class, you would go into a lot more depth than this. You would link this to propagation of brain signals through neurons. That's a whole other class. We won't be looking at that piece. So keep that in mind. This is only a sliver of information. It's just an example of how phosphorylation affects enzyme activity. So what are we looking at here is we're looking at the inside and the outside. And if you want to understand, this would be a process that's happening in neurons. And let's start up at the top. You see how we have the lipid bilayer. Then you have the green blob, which is the sodium potassium ATPase. And what's going to happen is that this is a great example of how ions can move through the lipid bilayer. But in this case, the sodium ion has to move against its gradient. So it's going to get pushed to the side where there's a higher concentration of sodium ion. And that will not be easy. It's going to require energy. So let's take a look at that. You have sodium ions come in to the binding site, shown here, and there's a phosphorylation event on the enzyme that creates a conformational change. Okay, and that conformational change, if you notice, the shape of the enzyme from here to here changes, and now the sodium ion gets on the outside. Okay, that over time is going to make the outside of the cell more positive because there's sodium out there. And then what's also helping that is the fact that now potassium ions bind to a special binding site for them. And what will happen is that the enzyme dephosphorylates. You see from here to here, phosphates released. And what that does is it pushes the potassium ion into the cell. And so what we're seeing here is, and I'll highlight it with color, two potassiums move in to the cell, opposite directions. So this is an antiport, right? Oops. Using that terminology, opposite direction. So 2 moles of potassium ion move in for every 3 sodium that goes out. And so what we're doing is changing the charge situation between the outside and the inside of the cell. On the outside, it's more positive. On the inside, becomes more negative. So this is one of our most famous examples of active transport where ATP is involved, phosphorylating the enzyme and allowing for a conformational change that pushes ions, in the case of the sodium ion, against its gradient. Okay, that requires energy, because what I mean by against its gradient, as more sodium gets pushed out, its concentration is going to increase on the outside, so we need energy to keep pushing it that direction. Okay, so here's the part that we don't go over, is how this is involved in neurons, being the signal sent down your neurons. And so for that, if you have more interest in that, you can look up, if you just Google how do neurons send their signal, it'll go in, it'll show this, and then it'll talk about other channels, which are called voltage gated channels. But that's a whole other discussion and a whole other course. Okay. More pictures. So a little bit closer up. The transporter binds 3 sodium from the cytosol, 3 go out, 2 potassiums come in. And so 3 sodiums from the cytosol, phosphorylation favors a conformational change, sodium ions release, dephosphorylation favors original conformation, potassium's released. And then on the outside of the cell, it ends up being more positive than the inside. Okay, again, showing the enzyme phosphorylated, conformational change kicks sodium out, dephosphorylation brings sodium in. And so this is just a great example of how phosphorylation events create a change in the enzyme's function. Okay, this is the kind of thing that I don't go into, but just put it on here as a slide. This maintains the electrical gradient, meaning the outside of the neurons with this pump going will be more positive than the inside, and that's going to help set up for signals being sent down the neurons, the brain signals. Okay. Okay. Another way that molecules move into a cell is through what's called receptor-mediated endocytosis. A great example of this will be LDL, which I know you've heard of with respect to cholesterol. We'll come back to this in a later discussion. So basically, the idea here is that the whatever it is that's coming into the cell is going to bind to a receptor, and that causes a change so that the receptor binds to some molecules called clathrates, and these clathrates start to polymerize, and then it causes a pinching in and formation of a vesicle. If none of that makes sense, that's okay, because we're going to look at the pic now. So you have your blue plasma membrane up here at the top. You have these receptors, and then there's a binding event. And then what happens is that there's a signaling that happens, and you end up forming what are called these Clathrin pits, and they form these circular structures ultimately that carry that particular ligand into the cell. So here's another picture. You have the cell membrane. You have the formation of these Clathrin-coated pits. Whatever has bound to it through those receptor and ligand binding, and then it forms these circular structures that carry your ligand into the cell. And what it does with it will depend on what it is. Later, when we talk about LDL and cholesterol, I'll give you a much better example, but we'll get to that part when we do fat metabolism. Okay, our last section, I think, is perhaps for us and where we're headed in terms of metabolism of glucose. This is really important. It's called receptor-mediated signal transduction. So I guess if I was to be honest with you, the endocytosis that we just looked at, the Clathrin pit discussion, I don't normally stress. It is an example, another example of how things get in the cell, and we will look at it later with the fat metabolism. But I think more important for us is this next part, which is receptor-mediated signal transduction. What this is that on the outside of the cell membrane, there will be a receptor. So in this picture, there's a receptor binding site or receptor, and it's going to bind a ligand which will be, in our example, a hormone. What a hormone is, the endocrine system responds to messages by the nervous system and synthesizes what are called chemical messengers. So I suppose in simplistic terms, a hormone is a chemical messenger. And it's going to be what's considered to be a primary messenger. And what is the binding event going to do is it's going to signal for something to happen inside of the cell. Okay, so let's take a look at the next slide. The hormones that signal the release of glucose from a cell, these two are very important players for us in biochemistry and in this course. So the two hormones that I'll focus a lot on, and something we'll take with us to the next section, are epinephrine and glucagon. And the difference between these two hormones depends on which tissue we're talking about. So here's that concept that different cells are different function. So they usually will have perhaps can have like a slightly different biochemical response. So in muscle cells, myocytes, the hormone that's going to cause the release of glucose from that cell is epinephrine. Okay. In the case of the liver, the hormone is glucagon. Okay. So, and let me just back up a bit. I know we haven't done this section. It's our next one. Is what do I mean? The release of glucose. It's not really, I kind of misspoke a second ago. The release is from glycogen. So in the muscle, glycogen is the storage of glucose, and we'll talk more about that next section, but it's going to break down into glucose that we can utilize for energy. Okay, in the liver, it's glucagon that will do that. Glycogen is also stored there and is mostly stored in the liver. Okay, now in the case of the liver, it may be used for energy. It may also be used to increase blood glucose levels, but we'll come back to that in our next section. Okay, so let's take a look at this here. The hormones, glucagon in the liver and epinephrine in the muscle cells are the hormone. And what they're going to do is they're going to bind to a receptor in their cell, on the cells. So the hormone goes through the blood, binds, let's say, to a muscle cell, and that receptor is going to recognize the hormone. They're going to complement each other. And then it's going to create some chemistry, biochemistry within the cell. So if I go back, signaling molecules can trigger a change in metabolism. It could cause an increase in what's called glycogenolysis, which was the release of glucose that I just described from glycogen. Okay. There can be other effects, not in the case of glucagon. There can be other effects too. So you could see a change in electrical charge across the plasma membrane. This would be more the neuron physiology that we're not going into. You could have a change in gene expression. But what I want to focus on for this example is number 1. Okay, that's the one that we're going to focus on. We'll come back to this in a second, and that. So what I want to get to, sorry, I do this a little bit differently in person, so that's why, bear with me here. Okay, so let's come back here then. When that hormone binds to the receptor, there's going to be one of the most famous of signaling pathways in biochemistry, which will involve something called G protein and phosphorylation cascade. Okay, so we're just focusing on number one, which would be, in our example, the hormone's going to be epinephrine in muscle or glucagon in the liver. Both of those will stimulate the storage of glucose to glucose that can be either used by the cell or put into the bloodstream. So we're just going to focus on this top one. These other two were just there to say that it's not specific to just that process. There's other things that a hormone can trigger, but we're just going to keep it simple with one type of example. Okay. What will be important to understand is that the binding of a hormone on a receptor will cause the formation of other molecules inside the cell that are called secondary messengers. For our example, the secondary messenger that I'm going to focus on is called cyclic AMP. These other two, inositol and calcium, are other secondary messengers, but I will not be focused on them. Okay. The interesting part about what's happening here is that when that hormone binds, so here's our hormone, epinephrine in muscle, or glucagon in the liver. The ultimate goal is to free up glucose. Okay, so that binding event is going to create a cascade of events on the inside. And one of them will be to produce from our hormone, that's our primary messenger, it's going to be to produce a secondary messenger. And then there's also going to be a huge number of chemical changes that goes on. Ultimately, the goal is to free up glucose. Okay, so there's a lot of details not shown here. That's just the big picture. So this is our big picture. And so let's brace for the specifics. Let's start with this one. If this is intimidating, I understand, because it kind of is. And then in the next picture, there'll be a handout that I actually like better than this pretty picture, but let's start with the nicer picture, I guess. We have the signaling molecule, the hormone. That's going to be, once again, epinephrine in the muscle or glucagon in the liver. Once again, the goal for those particular hormones is to free up glucose. We have a need for glucose, a need for energy. Okay, we come back to that part in metabolism. All right, now there's a big series of events that happen. There is what's called a G protein coupled receptor, so that's activated by the binding event. Okay, and this causes what's called a G protein to bind to a molecule called GTP. So you have an activated G protein. Okay, it's activated from this initial triggering event. Okay, and it's going to bind GTP. Now let me go back on the PowerPoint. Here's GTP. It should look like similar to ATP. This is ATP was adenosine triphosphate, GTP is guanine triphosphate. So the difference is really the nitrogenous base. That's the difference there. Okay, so that's where the difference between these two is that base. All right, now going back to the original here. Okay, so let's go back through it. Our hormone epinephrine binds. It creates an activation of this protein in the membrane. And what that does is it activates one of the most famous proteins in biochemistry called G protein. G protein will now bind GTP. And what it does is that activates another famous protein called adenylyl cyclase. And what does that protein do? Is it converts ATP to cyclic AMP, just a different version of it. It's going to have, it's not triphosphate anymore, it's monophosphate. It's a circular structure. Okay, so it promotes the formation of the secondary messenger. And what does the secondary messenger do? Is it's going to create what's called a phosphorylation cascade. And so this last part of this diagram, it's what's called a phosphorylation cascade. Ultimately, at the end of that cascade, glucose is going to get freed up. Okay, this is one of the most well-studied processes in biochemistry. I have done it described it to you in an introductory level. There's all sorts of details over in this part that I'm not going through, but that's not the goal of our course. If you're interested, you can go. There's numerous references online for all the nitty-gritties of that activation of the G protein alpha subunit here. I know that I've glossed over it, but that's because that's not the important part for us. The important part is understanding how the hormone comes into play to creating a signal for something to happen within the cell. Okay, now I know this is a really pretty slide, but I actually prefer this black and white slide, and I'll tell you why. Because there's a lot of information here. This does not show the original binding event of the hormone. It does not show the G protein activation, so it doesn't show that part, and then doesn't do that part. So we're coming in at the point where adenylate cyclase, that's an enzyme that is going to take ATP and convert it to cyclic AMP, our secondary messenger. Okay, the goal is to take glycogen, that's our storage of glucose, and free up glucose. You might be asking for what? Well, in the muscles, it's going to be for energy, it's for muscle contraction. In the liver, there's more options. It could be for energy. It could also be to put some of the glucose into the bloodstream if we have low blood sugar. Okay, good news, we go back over all that part later. Okay, so let's go back up then. Our hormone binds, G protein is activated, GTP binds to it, and in that phosphorylation event, adenylate cyclase is active. It converts ATP to cyclic AMP, and then things start to really happen. We have what are called kinases, and anytime you see the word kinase as an enzyme, it means that it's going to involve phosphorylation. Okay, so cyclic AMP is going to convert an inactive protein kinase A to an active one. So let me highlight. Here's the key, active enzyme. Okay, and here's where the body and biochemistry is so amazing. Protein kinase A is going to activate by phosphorylation events the activation of enzymes to free up glucose. Okay, so let's look at that side of the diagram. Protein kinase A is going to now be active. It's going to take another enzyme, phosphorylase kinase B, and activate it by a phosphorylation event. Now here, let's talk a little bit. See that OH that I've just highlighted in green? Okay. Think back to a previous slide. Remember threonine, serine, and tyrosine? Those have the OH. That's showing you one of those residues is being phosphorylated. Okay, so that is the serine, threonine, or tyrosine. Okay, depends on which enzyme it is. So we'll keep it more general. Okay, now phosphorylase kinase A is active. It's going to be the enzyme to take phosphorylase B, again showing this OH, right? Notice on the other side it's phosphorylated. Okay, and then it's going inactive phosphorylase B to an active phosphorylase A. Oops, I want to switch colors on that. Okay, and now we're at the end of our phosphorylase cascade, where the outcome is that the storage molecule of glucose called glycogen releases glucose. Now that is a lot of work, but there's an amazing thing that happens here is that you can have one event on the outside of the cell membrane. So one binding event on the outside of the cell create a major event within the cell. So the binding of one hormone can release thousands of glucose molecules. Okay, so this is a very, very dramatic process. You have a huge number of glucose molecules released from one binding event on the outside of the cell. And that's going to make it really effective. Okay. Now, there's one other thing on this diagram, is that let's think about something. If the body wants, the cell needs to release glucose for energy, then it's going to go through this process from the original hormone, epinephrine, for example, in the muscles. That's going to release the glucose inside of the muscle cell free it up for energy. Okay. Now, it would not be effective if at the same time that we're trying to release glucose, we're still trying to store it. So over here, what we're going to see, and I'll pick a color here, is at the same time, this branch, the blue branch that I'm showing, okay, this is going to deactivate the enzymes that store glucose. So you see over here, we're activating enzymes to free up glucose. On this branch, protein kinase A will deactivate the enzymes that store it, because you want to shut that process down. So this is activation over here, and this is deactivation over here. So protein kinase A will deactivate an enzyme called glycogen synthesis of glycogen. It's going to take that one from being active and deactivate it. So this is really an amazing process, if you ask me, because you have the one hormone binding on the outside, goes through a series of conformational changes in those enzymes, forming protein kinase, activating protein kinase A, and protein kinase A has two functions. It's going to activate the key enzyme that gets the process going on this side to release glucose, but at the same time deactivates the enzyme that stores glucose. Okay, I will not be asking you to, to draw this by memory, but when you see it in your homework, you'll see the kinds of questions that I ask. Like it will ask you, what is cyclic AMP? That's a secondary messenger. What's the hormone involved? Who are the hormones that are involved? What's the role of phosphorylation? Well, phosphorylation can activate or deactivate enzymes. Okay, so epinephrine and glucagon, these are the physiological effects communicated by these primary messengers. So these are the primary, right? They're the hormone. But they both trigger that G protein event that creates the secondary messenger. We just went through the idea that in muscle cells, glucose is freed up. We also talked about in the liver, glucagon, glucose is freed up. Okay. Now, if you notice, other things can happen too. So epinephrine in fat cells will cause the formation of those triacylglycerols we practice drawing. In the heart, increase heart rate and smooth muscle relaxation, calcium release. So there's other physiological effects. I only focused on two of them. So the two that I focused on were these, this one and this one on the previous slide. Okay. Later in the course, we're going to look at triacylglycerol degradation. And so we're going to look at these, but that's a little bit later in the class. Okay, so right now, obviously, what's dominating our lives is the discussion on viruses. So viruses have to get into a cell. And so this is a very scary picture, but it just kind of want to show you that there's are different viruses and how are they going to get into the cell to wreak havoc. And so this shows you that they do so through receptors. And this is going to be, so here's our HIV virus. So there's HIV. Here's adenovirus. Oh, here's the flu virus. Okay. Obviously we're looking at the coronaviruses, but a virus has to find its way inside of our cells. And what they want to do is they want to use the cell machinery inside of the cell to replicate. Okay, now that's a whole other branch of biology and biochemistry. But since I think it's so critical right now, I'm going to be having another video. I'm going to post up a video. It won't be mine, but it'll be one that is already has a discussion regarding HIV. And that'll be part of a worksheet. Originally, the worksheet was going to be due next week. I think I'm going to open it up and make it could be due towards the end of the semester to give you a little bit more time on this. But I just want you to be aware that viruses want to enter the cell, and they're going to do so by binding to receptors on the outside of the cell. So one of the primary ways that we want to stop viruses is by blocking their ability to bind to those receptors. And that's where some of the drug treatments come in. Okay, so stay tuned for more on that. Okay, so I know this is a really difficult section going back to here. I know this is really scary, but we'll talk more about it during Zoom on Thursday and probably Tuesday of next week. So we have some time. I just want you to watch it and try to understand the general principles we're trying to learn, which is how do molecules get through the cell membrane and how do signals get sent through the cell membrane.